Blood is a transport tissue that has many functions in our body, not just carrying oxygen around our body. So a number of functions. We do have transport, not just oxygen. We do have oxygen. Carbon dioxide is a waste gas. We've got urea. We've got hormones. We've got lots of proteins in there, like fibrinogen, which is to do with blood clotting. Products of the digestive system, like glucose, that need delivering around the body, that will all get transported in the blood. Also transports heat. Now, we thermoregulate to 37 degrees Celsius, but the extremities of our body, especially on a cold day, are not going to be in an environment where we're at 37 degrees. So we can transport heat in our blood to the extremities of our body to maintain our temperature. We've got a defense role as well against infections from microorganisms, bacteria, parasites, viruses as well. Our blood is very good at protecting us from these things too. We have a filtration role of blood as well. So in our kidneys, we are going to be filtering using high blood pressure in our kidneys. And we'll talk about this more in a later topic, but that's an important function of blood. And a final function to remember is homeostasis. So this is maintaining a balance, a set point. And again, we'll talk about this in later weeks. But for example, the pH of our blood is maintained by our blood. It's got this role in our body. If we look at what blood is made up of, it's got four main components. We have plasma, we have erythrocytes, also known as red blood cells, and we have platelets, and we have white blood cells, also known as leukocytes. So there's lots of different things. So we've got plasma, red blood cells, platelets, and white blood cells as well. Now this cartoon here is what happens if you put blood through a centrifuge. So this is a machine that will spin very fast and will sort by density. So you end up with heavier red blood cells at the bottom, and plasma is the liquid part, so that's lighter. This buffy coat is the layer where the white blood cells and the platelets separate out as well. In terms of how many of these things we have, about 55% of our blood is the plasma, about 45% is erythrocytes, and less than 1% is the platelets and the white blood cells. But if we look at 1 milliliter of blood and the numbers of these things that we might have, on average, we will have about 5 million red blood cells. Now, sometimes this can be higher. So if you live at altitude, your red blood cell count will be higher. And we'll talk more about why that is in a later topic. You will have about 7,000 white blood cells in a milliliter of blood. It's a tiny little drop of blood. And approximately 250,000 platelets. So huge amounts of cells in even just a very small amount of blood. So we will talk about the different components in turn. So plasma is the transport medium. It's the liquid part. And it has lots of things dissolved in there. And we mentioned some of them before, but you have proteins dissolved in like fibrinogen, you'll have carbon dioxide in there, sugars, salts, ions, all dissolved in there. It's a pale yellow colour. What you've got here, this image is showing you frozen, fresh plasma. So this is when people donate blood. Sometimes it is kept as whole blood, ready to put the whole blood, all the blood into a person who needs a transfusion. Sometimes people just need plasma. So in the blood donation service, they will spin the blood, separate the components so they can be used as efficiently for patients as possible. Like we said, 55% of the blood is made up of plasma, and it's mostly water. Involved in keeping pH constant, so it's a buffer solution, which is part of the homeostasis part as well. So basically, anything that can dissolve in the plasma is easy to transport around the body. About 45% of our blood is made up of red blood cells, also known as erythrocytes. Cyte means cell, so anything that has cyte at the end tells you it's something to do with a cell. So these are the erythrocytes. You've got a image taken here. This is taken by just a light microscope. It's got a stain on it as well, and you've got all sorts of different things shown on here. So these structures are the red blood cells. You've got some white blood cells here, some leukocytes, so they are a bit bigger, and you've also got some platelets here. These are the little cell fragments, and we'll talk about what they do in a while. Like I said before, you've got about 5 million red blood cells in every millimeter of blood. And the red blood cells contain hemoglobin, and hemoglobin is the respiratory pigment that carries oxygen around our body. Red blood cells are made by a process called erythropoiesis, which is very hard to say and even harder to spell. Erythropoiesis. So you've got the erythro part, which tells you it's something to do with red blood cells, and poiesis, erythropoiesis. And in adults, in us, red blood cells are made in the bone marrow. It's in the red bone marrow inside some bones. Now, the bones it's made in, and you often see pictures of like the femur or the long bones. It's not where it's made. Red blood cells are made in the vertebra that make up the spine, the spinal cord, in your ribs, inside your sternum, your breastbone, the skull, and the pelvis. So these are the bones in which red blood cells are made, and we make a lot of them. So we make about 2 million per second, which kind of blows my mind, if I'm honest. A fetus during pregnancy is making red blood cells in its liver, so different in a fetus, and the hemoglobin that is in a fetus is different to adult hemoglobin, and we'll talk about that more when we talk about gas exchange later in the semester. Red blood cells last for about 120 days, and then they need to be destroyed and recycled, and they are broken down in the liver, so this big organ here, and also in the spleen, which is shown in purple here. So they will be broken down and recycled. But because you're making 2 million per second, if you do donate blood, you'll be replenishing your blood volume within a week at the most. Red blood cells have a very classic structure. This image here is taken using an electron microscope, and actually this is a scanning electron microscope image. Scanning, it looks across the surface. So you've got your red blood cell here. They're very small. They're about 7 micrometers across. So the sign for micrometers is like this. So that's 7/1000th of a millimeter. So incredibly small, about the same diameter as a capillary. And we'll come on to that more when we talk about blood vessels and also when we talk about gas exchange. But being about the same size as a capillary lumen means that they can squeeze through in nice close contact, so you've got a faster rate of diffusion for gas exchange. Very flexible membrane, so they're not going to get stuck. They squeeze through the capillary, so we've got nice even blood flow. They're a cell, but they don't have a nucleus. So they don't need a nucleus. They're not going to use the DNA to do anything. And if they don't have a nucleus, it means they've got more space to put hemoglobin in. So they've got more space to carry the respiratory pigment and carry oxygen around their body. They have a classic shape called a biconcave disc shape. It's like a donut, a ring donut, but without a hole the whole way through. So it's like this shape. So it's a classic shape. And you can see that on the image here. You've got like the dimple in the middle here. And this shape means you've got a nice large surface area to volume ratio. So it means it's faster for gas exchange. These images are also taken by scanning electron microscope. I really like electron microscope images. But what we've got here, we've got a normal, healthy red blood cell, but we've also got a red blood cell from someone suffering from sickle cell anemia. And we'll talk more about sickle cell anemia throughout our course together. But this is the one with sickle cell anemia, and this is a normal red blood cell. Hopefully you can see the sickle cell red blood cell is longer, thinner, it's got sharp pointy ends, and it means that when it's traveling through capillaries in the extremities, certainly it struggles to get through the capillaries and can get stuck, and this can cause problems with carrying enough oxygen around the body, and also periods of intense pain when people who have sickle cell anemia have what they call a crisis due to this, and they get incredible amounts of pain. It's worth mentioning that these images that are taken by scanning electron microscope are usually grayscale, so they're usually black, white, grey, and these images have had false colour added to them. And you'll see that sometimes with electron microscope images, but anytime you see an electron microscope image with colour on it, that's been added afterwards using software. Because electron microscopes use electrons to form the image, they're not going to be able to produce a colour image. White blood cells are also known as leukocytes, and you've got an image of two white blood cells here. So this is a white blood cell and this is a white blood cell, and they are involved in our immune system. They do have a nucleus, so you can see we've got a nucleus here and here as well, larger than red blood cells, which you can see from this image as well, and a lot of them have got a very short lifespan. So they might live for only a few hours or a few days. And if you think about how many infections you're potentially exposed to in a day, your white blood cells can mount a very fast immune response, but then once that risk goes away, they don't need to be producing those particular cells again. You do have other cells, memory B cells, for example, which will form more of a more long-term immune protection response. But mostly they're quite short-lived. Now there's a lot of details about white blood cells on this slide, and you do not need to know all these details. It's beyond the scope of what we're looking at on this course. It's worthwhile just pointing out a few little things. So this is showing you how these blood cells are made. So it's showing you how red blood cells are made. There's your erythrocytes there, one of the blood products. Thrombocytes, that's another word for a platelet. So platelets are shown here. And everything else is the white blood cells down here. All these other things are white blood cells. They're involved in protection. Many of them have what we call phagocytic properties. This means they can do phagocytosis. Phago means eating, and cyto is cell. So they eat cells. So something like a macrophage, if you are exposed to an infection, a bacterial infection, for example, the macrophage will surround that bacteria, bring it into the macrophage using endocytosis, and then take it to the lysozyme, as part of the cell structure and destroy that bacteria to protect you. So a lot of these structures are phagocytic. So neutrophils are phagocytic, and monocytes, macrophages, are phagocytic. This part here, the B cells and the plasma cells, these are involved in making antibodies. So once you've been infected with something, your body will make antibodies to clear the infection, and then you'll keep some antibodies, keep some of these cells present so that if you get infected again, you are mounting an immune response. So when you're making a vaccine, you're trying to activate this response. It's called the humoral response, in order to protect you against future infections. T lymphocytes, T cells, are involved in destroying infected cells. So if you've got one of your cells is infected with a virus, for example, T lymphocytes can be involved as part of a cellular response in destroying that. But like I said, far more details than we need to know at this level. It's just enough to remember that white blood cells are there to help protect against infection, and look out for immunology at university is absolutely fascinating. Platelets are also known as thrombocytes, and these are little cell fragments. So they're not a whole cell. If you remember to the previous slide, they kind of come off megakaryocytes when they break up into little pieces. They don't have a nucleus, and you've got an image of them here. So we've got a red blood cell, we've got a white blood cell, and this is a platelet. And this is again taken by electron microscope, a scanning electron microscope, and you can hopefully see that the platelet is a cell fragment. And in your plasma, if a blood vessel is damaged, they are going to start blood clotting mechanism to make sure that you don't keep bleeding, and also to make sure that infection doesn't get into your blood system. Now the blood clotting process, you've got an image of the process in your topic book. Far more details than we need to go into at this level. What's important to remember is that the platelets, they start a chemical reaction, and they start this chemical reaction, and what they'll do is they'll take soluble fibrinogen, which is dissolved in the plasma, and they turn it into an insoluble form called fibrin. So you've got a broken blood vessel up here, the platelets come in, start this cascade of reactions, and the fibrin comes in and sticks into the hole. And when the fibrin, which is now insoluble, sticks in this gap, it sticks loads of red blood cells together as well. And white blood cells will move in as well, and it forms a clot to stop the blood, so the blood doesn't keep escaping and stop infection getting in. If you've ever cut yourself and you end up with, we call it a scab in England, when you get that crusty surface on your skin, that is you've activated the clotting process, and underneath you are healing. You've got the fibrin holding the red blood cells and the white blood cells all together and healing and making new cell skin to replace the damage. And you, in the process of that, you'll be repairing the blood vessels as well. Beautiful image of a blood clot. And on the fibrin here. This is scanning electron microscope again. This is how it would look when you take the real image. So this is the grayscale image that you would see. What you've got in here, this is a red blood cell. So all the things that look like this or like this, a red blood cell. All these fibers forming this mesh, kind of mesh structure over it. This is the insoluble fibrin, and that holds this blood clot together. Normally, I prefer grayscale electron microscope images, but this is one that's had false color added to it, and I actually quite like this image. So it's much easier to see the red blood cells because they've painted them red. What you've got right in the middle here, right here, this is a white blood cell. So that is there to clear up any infection that might have happened once you opened up that blood vessel. And then all of these fibers here, this is the insoluble fibrin that we talked about that is involved in forming this structure. In our next session, we are going to talk about the blood vessels. So these are the tubes in which the blood will travel. Thank you.